Gotta get the momentum. Okay, today we're going to talk about ticks and Rickettsia. So, I'm an expert on Rickettsia. I'm so-so on ticks. So this is a fun lecture, at least for me, in the Rickettsia. Oh, you don't have a laser pointer. At least in the sense of the Rickettsia stuff. And I'll just say that Rickettsia are bacteria. We'll talk a lot about them. There's one Rickettsia we won't talk about, which is Rickettsia prausechii, which causes typhus. We have a whole separate lecture on typhus. Today we're going to talk about ticks and then Rickettsia, everything that's not prausechii. Okay, so ticks. This is a phylogeny of arthropods, so arthropoda. So everything in here are arthropods. This is an outgroup. So things like horseshoe crabs, lobsters, crabs, myriapods, which are like centipedes, millipedes, and then insects over here, all those are arthropods. So arthropods are segmented molting organisms. So segmented, they have segments, and they molt means they have a extra cuticle, like a hard shell. And so to grow, they need to, I already explained this once, they need to shed that extra skin so that they can grow larger. So that's what molting. So all these are segmented molting organisms. Did I get that right, Basu? Okay. Basu's double checking me. Okay, so insects, what we normally think of as insects are actually hexapoda. So six-legged things are insects. Now ticks are not within hexapoda. They're not six-legged, and they're not really even they're not too closely related to insects. They're more over here, more closely related to spiders and crabs. So chelicerates, and this was a bonus question on the first test, are really what we're going to start focusing in on now. So a little bit more about that. Within chelicerates, so they're separated by their mouth parts, and we'll move on that. But then also they have 8 legs. So arachnids have 8 legs, and ticks have 8 legs. So they're considered within arachnids. And then the subclass is Acari, which is mites and ticks. So let's see, what else is unique from this slide? Here's the chelicerates. Ticks transmit the widest variety of pathogens. So they transmit a lot of different things, viruses, bacteria, protists, diseases, and they're feeding on many different things, and they take multiple blood meals. So it allows them to kind of accumulate all these dirty things inside their system, which they can transmit. But they don't affect as many quantitatively people as mosquitoes. And I kind of talked about this in the sense that, like, we're really afraid of mosquitoes more and things that fly because they spread a lot more easily. Whereas ticks are walking organisms, and you mostly really only get you encounter ticks if you kind of invade their territory. It's like sharks. You don't have to worry about getting bit by a shark if you're not in the water. So most of the time when we get diseases from ticks, it's because we're out in the field tromping around and we get a tick. So ticks are arachnids. I already said this. They have chelicerae fangs. And the difference between chelicerates is that when we talked about the structure of the mouth parts, there are mandibles, okay? And chelicerates don't have mandibles. They have chelicerates, chelicerae. So these are the chelicerae. Sometimes the chelicerae have venom glands. So like if the spider bites you and injects you with some poison, what it's hitting you with are its chelicerates or chelicerae. Is that correct? Chelicerae? That's the plural. I think that's the plural of how you say the two chelicerae, and chelicerates is the taxonomic clade. Okay, so within the larger group, there's spiders, okay? And the mites and the ticks are really closely related to spiders. So again, they're within arachnida, within the class. And the subclass is Acari. So when you hear Acari, that's people who study mites and ticks. And there are people who just specifically specialize in mites and people who specialize in ticks. And there's many different species of mites, many more mites than there are ticks. Oftentimes you can find mites that live off a particular organism. So like there's mites that eat Drosophila eggs, and there's mites that live on X bee organism or whatever. So there's different kinds of mites because many of them are ectoparasites. And we eventually will talk more about mites because there's some diseases that might spread, but for now we'll focus in on the ticks. So ticks are basically large mites, and some are important vectors of human diseases. So most of you have already heard of Lyme disease. That's kind of what everybody thinks of when they think of tick-borne diseases. We have a whole lecture on Lyme disease, but that's caused by it's caused by the pathogen Borrelia burgdorferi. And the ticks pick up the bacteria just like mosquitoes. They have different life stages, and each of those life stages actually has a blood meal. So they're picking up these bacteria or these viruses when they're feeding on their hosts that they take a blood meal from. And then if that tick comes and bites you, you get a disease. So it's very similar. It's a vector. It's the same kind of concepts as you would encounter in a mosquito. Okay, so the life cycle of ticks. Ticks go through 4 life stages, and they're kind of unique. So you technically, you'd say that they're not holometabolists because they don't go through metamorphosis. But I don't think you can actually say that they're hemimetabolists because hemimetabula is actually like a group of insects, and ticks aren't insects. But their life cycle looks like hemimetabolous insects in that they molt, and their molts look pretty much the same organism, so they don't change the structure of their their being, like metamorphosizing insects do. But they get bigger with each stage. The one big thing that's different that you want to know that's unique is the larval stage of ticks only has 6 legs. So if you see a larvae, it's microscopic. It's very, very tiny. It'll be hard for you to see it. But if you look at it, it only has 6 legs. And this is also true for the mites that we're going to talk about later. So that's where some people might get confused, is if they're looking at a larva and then they might think it's an insect because it has 6 legs. But actually, you want to know that the larvae of ticks and mites have 6 legs. And then in the next stage, the nymph comes out with the 8 legs, and then you get an adult with 8 legs. Let's see. So all stages of ticks, this is kind of unique to ticks, all stages take a blood meal. So as soon as it comes out of the egg, the larvae will take a blood meal, the nymph will take a blood meal, and the adult will take a blood meal. So there's 3 blood meals to complete the tick life cycle, rather than in the mosquito, there's only 1 blood meal. So again, this means that they pick up a lot of diverse stuff because it takes them a lot more blood meals to complete their life cycle. Let's see. So if the larvae, if you get bit, if you find a tick on you that has 6 legs, you don't have to worry that you're going to get a disease because it hasn't blood fed before. Does that make sense? So if you get a larvae on you, you don't have to worry too much. Unless there was a maternally transmitted disease, which can happen. But for the most part, if you get a larvae, you don't have to worry. What we most worry about are the nymphs and the adults. Let's see, 3 blood meal, not whole. I'm looking at my notes to make sure I got everything. And you'll be shocked to see, when I first saw the first tick nymph I've ever seen, it was so tiny, it's shocking. Like, when you look at this, it's kind of like, oh, I can see that. But like when I first saw my first tick nymph, I saw it on my son, and I thought it was literally like a speck of dust. And then later, I saw it at the same spot the next day, and then I looked in closer, and I saw it was an engorged tick nymph. So, like, they're so tiny that it's really difficult to see them. Okay, tick mating. So the way that they mate is, so some insects will like swarm and mate. So the reason, kind of like one of the reasons that the males blood feed. So that's another thing, is both males and females take blood meals. So this is different from mosquitoes and a lot of the dipterans where only the female will take a blood meal. In ticks, male and female both take blood meals. Okay, and one of the reasons that the males take blood meals is because they need to be in close proximity of the female, right? Because they don't fly. So they need to be like geographically in the same space that the females are in. So they have this host seeking and feeding behavior because mating actually occurs on the host or in the nest. So if you find a female on a mouse or something, oftentimes you can find a male who is also on that host, and they will find each other on that host to mate. And the females will secrete a sex pheromone, 2,6-dichlorophenol. You can actually buy this from Sigma Aldrich if you want to spread tick mating pheromone all over yourself. It's only $27. So this is you don't need to know the chemical structure. You don't even need to know the name. But know the concept that if they're on a host, a female will secrete this mating pheromone, which will attract the male, and the male will then find the female and mate. So the mating is really weird. And in this case, it's mating with an engorged female. So it's so weird to see the engorged female because their size changes so drastically. So this is the female. And you can see her legs coming off here. And this is the male. And so the male, and you'll hear about this kind of stuff in other insects too if you study insects. So the male will like release the spermatophore, which is like a ball of like sperm and I guess nutrients for the sperm, whatever. And then the tick will actually like pick it up, okay, with its mouth parts, and then it puts its mouth parts into the female vulva. So that's how that they mate. And then molting occurs off the host. Okay, so I say show the 4,000 egg picture. It's pretty crazy. I saw this picture. This is in the textbook, and it's pretty crazy. So we've talked a little bit about this, where there's different strategies for insects. So there's K and R strategies. K is the carrying capacity, is what Basu said, capacity. And insects that have a K strategy, or insects or arthropods, make thousands of babies. Correct, right?
I didn't know.
Or is it reverse?
It's reverse.
It's R?
Any organism that has K strategies, I think they are larger animals, so they produce fewer number of offspring.
Fewer. Okay.
And do care more on that.
Okay.
Whereas
right.
other strategies, they produce larger number of offspring.
See, I was testing you. Okay, so I'm testing you. So the R strategies produce thousands. So oh yeah, see I have here R. High R, low K. Yes, that's what Basu said. Okay, so ticks are definitely they produce thousands. And so this is a picture of a tick that's laying 4,000 eggs. So they have a crazy ability to make many, many, many eggs. And if you think about it, they want to make lots of eggs because their life cycle is very hard to complete. They have to find 3 different hosts and take a blood meal successfully 3 different times and survive through that to complete their life cycle. So it's hard for them to do that. So they need definitely lots of eggs. Let's see. After the female lays the eggs, she dies. Okay, so it's not like a mosquito where a mosquito will take a blood meal, lay some eggs, take a blood meal, a second blood meal, lay some more eggs, take a third blood meal, lay some more eggs until she dies. It's not like that. The ticks will the adults will take one successful blood meal, lay their eggs, and die. Okay, so tick rearing. So studying ticks in the lab is notoriously difficult. It's hard to rear ticks, and you'll see why. One of the reasons is so if you have to figure out schemes to feed them. So some people will membrane feed them, where they rig up a little apparatus that runs blood through it, and then you put a membrane, and then the ticks can come on and feed through the membrane. But some ticks don't like to do that. So some ticks you need to like set up like a shaved mouse or like a shaved rabbit, and you set up this little arena, and then you have to like put the ticks in there, and then they feed. So some people will feed them on hamsters. At University of Minnesota, they would do it on mice. Nobody here is rearing ticks, but they're notoriously difficult to rear, and it takes a lot of time. let's see. One of the reasons is because their life cycle is 2 years long. So it's not like a mosquito, where a mosquito's life cycle is like 2 weeks. The tick is in it for the long haul, 2 years. Okay? So the eggs will hatch in the spring. Okay? And then it's seasonal. So then the larvae will come out in the summer and they'll find a host. They'll take a blood meal and then they'll just chill out, okay, for the fall and the winter. And then they go into a hibernation, like a diapause. And if there's lots of snow, that actually helps the ticks because the snow actually insulates them and keeps them warm in their little microclimate. So if you have thick snow layers, oftentimes you'll have huge, massive tick seasons the next year because it actually helps them. Okay? Then the nymphs will come out in the spring. So they'll do their molt, they'll come out, they'll find a next host. And then the larvae is usually, so like in Lyme disease, the larvae is usually what will feed in, will pick up the Borrelia burgdorferi, the Lyme disease causing pathogen. And then if a nymph finds you, a human, that's usually when you'll get the disease. But their preferred host is things like deer or large vertebrates. Okay? Then they'll take another blood meal, they'll molt to adults, and then sometimes they'll lay their eggs right then, or they will oftentimes diapause through the winter underneath the snow and then lay their eggs. So this is their life cycle. It's a very long life cycle. Some can do it all in a year, but most are usually 2 years. So 1 to 2 years, depending on the species, depending on the geography, the climate, things like that. That will all impact the life cycle. Let's see, risk of infection is high. This just notes that what you'll have, and this is actually how they originally found Lyme disease, is they noted that the disease, there were a bunch of people coming into the clinic at a certain time of year, only in the spring. So that's a sure sign of a tick-borne disease, because that's when the nymphs are out questing, looking for that blood meal. Whereas in the fall or the summer, the nymphs aren't doing that anymore, or especially in the winter. So if you see a disease that pops up in the spring periodically, yearly, that's a sure sign of something like a tick-borne disease. So that's one of the reasons how they actually originally discovered Lyme disease. Okay. So host seeking. How do they find their host? What do they look for? More on this in a bit. Some of them will crawl onto the host, and once they're on you, they'll find a feeding site. So they like warm, moist areas. So they'll go to your armpits. I got Lyme disease, and it was actually a nymph that was in my armpit that I was not even aware of it. So they'll go to places where you can't see, which is kind of scary, and then they will insert their hypostome. We've seen this picture before. This is what their hypostome looks like. So they're hematophagous, they suck blood, and they have these serrated hypostomes which insert and then make it like a fish hook so you can't pull it out. So it's a barbed hypostome, and then they will secrete their saliva, which has cementing proteins, anticoagulants, and that's when they vector their diseases. Okay, so there's two ecological strategies for how the ticks feed, how they find their hosts. One is there are ambush ticks. So they will literally climb up to the top of a long piece of grass, and they will put out their wings, or not their wings, but their legs, okay? And their legs are just waiting for somebody to walk by, and then they grab on. So it's gets brushed. And then there are hunter ticks, which will actively they smell for stuff in their area, and then they will crawl to find that host, and they will crawl on you. So there's different strategies that they use to find their host. And the host selection, just like any insect or any parasite, can be highly specialized. So there might be some tick that really can only take a blood meal on a particular mouse, because maybe that's the only mouse that's running through the little areas where they're hanging out on the grass, and they specifically evolve to have close host associations. Okay? So they're not always they're not generalists in the sense that they can specialize in the organism that they take the blood meal from. So they're famous for when they take the blood meal, their body weight can change 100-fold. So these are like the biggest engorging organisms, I guess, in arthropoda. I think I can say that accurately. And we talked about in the feeding lecture that ticks actually, when they're blood feeding, will actually have to actively synthesize more cuticle to actually hold the blood meal. So they actually have to, as they're feeding, make themselves bigger to get as much as they can. Yep, so I say that here. Some hard ticks literally synthesize new cuticle as they feed. Okay, so there are soft ticks and there are hard ticks. And we'll get into kind of like the families of ticks. Do I want to say that yet? No. But the soft ticks they don't have like that you think of. So if you look at this picture, hard ticks have like this hard dorsal shield, or the scutum. So it's like a hard shield. Soft ticks don't have that. And soft ticks, you can't see their mouth parts. Hard ticks, you can see their mouth parts. So this is a soft tick, you can't see its mouth part, and it kind of looks soft and squishy. Okay? So there's hard ticks and there's soft ticks. The soft ticks are mostly on animals. So like bats and birds. And so the soft ticks, they spread some diseases to humans, but we're not generally worried about them as much because we don't come into contact with them. Mostly what they do is they will be on like a bird or whatever, and then they'll take a blood meal, and then they quickly fall off. So they take a blood meal very quickly. They fall off into like the nest, and then they will molt in the nest. They won't stay on the host. So in contrast, hard ticks, these ones, which are the ones that spread diseases, they will stay on that host for days consuming blood, okay? There's just a note here. You're unlikely to see soft ticks just because most of them are, again, on like birds, bats, things like that. Most of what we see are the hard ticks. Let's see. Okay, so soft ticks versus hard ticks. These are characteristic hard ticks. Okay, so the family is Ixodidae. These are the ones we care about. You can see their mouth parts, so we know they're hard ticks. Okay, so let's talk just quick about the three families. So there's just two kind of that you just want to know what they are. So if you see Ixodidae, that's all hard ticks. Okay, if you see Argacidae, that's all soft ticks. These guys. Okay, so they're literally, the families are separated. Hard ticks, soft ticks. Hard ticks are Ixodidae. Argacids are the soft ticks. Okay, and then there's a third family, which there's only one type specimen, Nataliella namaqua. Okay, so I'm just putting that in there so that you know there's three families, but I'm not going to ask you anything about that because we don't worry about that one. But there's three families, but you basically just want to know hard ticks and soft ticks. Okay? Doesn't it look delicious? Okay, so let's talk about the important genera. So ticks are very easy to just distinguish by eye. It's not difficult to know the genera, and they're very easy to key out. So if you ever find a tick and you want to know what it is, just go to Google and just search key of ticks in Alabama, and it's very easy to key them out by eye. So Ixodes ticks, they don't have eyes, they lack eyes. These are the deer ticks. Okay, so when you hear deer tick, when you hear Lyme disease, these are Ixodes ticks. Okay, so not to be confused with Ixodidae, which is the family of hard ticks, that's everything. Ixodes are the genera that are the deer ticks. Okay, they spread Lyme disease, anaplasmosis, babesiosis. Okay, there are dermacenter ticks. So Dermacenter variabilis is the American dog tick. So if you are a veterinarian, you want to know about this. There's Dermacenter andersoni, which is the Rocky Mountain wood tick. These are famous for transmitting Rocky Mountain spotted fever and anaplasmosis. Okay, and then there's Amblyoma. Amblyoma are ticks that have eyes. Okay, these transmit ehrlichiosis. So you hear the Lone Star tick. This is what this looks like. It looks like, I guess, it doesn't really look like that, but it looks like there's a star on the back. That's why they call this the Lone Star tick. And then there are Ripocephalus. So there's another dog tick. This is the Ripocephalus. And this can infest houses if it's on your dog, so this will distress humans. I don't know if we have time. If we have time to tell you my story, ask me at the end of the period. I can tell you my story about ticks. So Ripocephalus can vector Rickettsia, Connerii, Ehrlichia. We'll talk more about those. If you're a veterinarian for agriculture, Ripocephalus microplus is an important livestock tick, which transmits cattle fever, okay, or bovine babesiosis, or Anaplasma marginale, which is a big issue in cows. So if you care about cows, this is a tick you want to know what it is. And then there is Hyaloma, which transmits Crimean Congo hemorrhagic fever. So those are kind of the important genera. You want to have just kind of like heard of what they are. And if you want to be able to tell them apart, like I said, it's very easy. They're just visually different. Very easy to tell them apart. And it's very easy to tell male and female apart because the male and female are clearly sexually dimorphic. So they're completely different. So it's very easy to tell what they are. So the things that you're going to be looking at are not only, so you want to think about where you found the tick, okay? So like when I got Lyme disease, it was easy for me to identify the tick, even if I wasn't able to look at it, just because I know I'm in Connecticut, which is like deer tick, Lyme disease territory. So you kind of want to think about where you are, what habitats you're walking through. That will help you identify the tick. And then once you have it, you can just look at it. What things you want to look at are the patterns on the scutum, which are the visual things, and I'll show you. And then the mouth part length. So the ones that you might actually confuse are amblyoma versus dermacenter. Sometimes these can kind of look similar, and then you just look at their mouth parts. So amblyoma have long mouth parts, and dermacenter have short, stubby mouth parts. Okay, those are the only ones you might actually like confuse. Okay, so just to show you how easy it is. Here's Ixodes scapularis. So this is the deer tick that will give you Lyme disease. The females, easy to tell because they're orange. Okay, the males are not orange, and they're basically black circle things. Okay, here's amblyoma. Okay, so this is the Lone Star. So the Lone Star tick will have this little white spot on the scutum. And then this is where you can kind of get them confused. This male kind of looks like a dermacenter. But again, you look at their mouth parts. So in amblyoma, the mouth parts are longer. And if you look at dermacenter, their mouth parts are shorter. So very easy to tell. Anything else unique about this? You can see here clearly that the larvae have 6 legs, whereas everything else have 8 legs. Here are dermacenter. So these are the like Rocky Mountain. Well, this is the American dog tick. So they look like this. I don't know how you describe that, but it's white, brown patterns, and clearly the female is very different from the male. Maybe you would get this confused because it kind of looks maybe in some conditions of lighting this might look kind of orange, and you might confuse this with this. But if you really look at it closely and you have a key, it's very easy to tell. So basically, what I say. Wait, do I have anything else about Ripocephalus? No, basically what I say is, if you want to know what it is, male, female, what it is, literally just like Google key for ticks in Alabama. And it's very easy to just look because they're so obviously sexually dimorphic and they're so different from each other. So it's easy to tell. And this is how you can, if you ever find a tick on you, you can tell, well, I might be worried about this disease. If you key out the tick, you know that if it's a deer tick, you might have to worry about Lyme disease. If it's a dermacenter tick, you might have to worry about Rocky Mountain spotted fever. So if you find a tick on you or a dog, use the opportunity to key it out, figure out what it is, and use the opportunity to think about what diseases you might have just that might have just been transmitted to you or your dog. And also, if you get a tick, you save it, right? So, because oftentimes you might not see symptoms right away if you get sick. So you save ticks that you find on you or your dogs. You just put them in a bag, put them in the freezer, or put them in a little bottle, whatever you want, but save them. And then if there's ever a problem, you can be like, nope, this is the tick. And then people can just PCR the tick, figure out if it had a disease or not. The problem is that people pick off ticks and they throw them in the garbage, and then they get a disease, and like there's no way to really tell like what happened. So just save the ticks. It's easy, and it's kind of cool, kind of fun. Okay. So let's talk about how they transmit diseases and why they're such fantastic vectors. Okay. So again, this is different from mosquitoes, different from all the other vectors that we've looked at before. Ticks feed very, very slowly, okay? So they come on you, they cement their face into your skin, and they stay on you for days, okay? And they're constantly sucking blood, okay? And they actually alternate. This is why they transmit so many diseases, because they'll spend long periods of time sucking up blood, and then they'll spend long periods of time straight up spitting into you, spitting all their enzymes, spitting all their diseases into you. So they alternate, okay? At points they're sucking, and at points they're spitting. And for their days, the days long that they're doing this, okay? Now, because ticks feed like this, the diseases that they spread, you're more likely to get a disease from the tick if it's been on you for longer time, okay? So if you find a tick that is not engorged, that's just on you, just inserted its mouthpiece, you're probably not going to get a disease because you caught it early enough. It didn't have enough time to be sucking blood and spitting stuff out and sucking blood and spitting stuff out. If you catch a tick questing on you, so that means crawling on you, you don't have to worry about a disease because it didn't blood feed yet, okay? If it blood feeds, you'll know it because it will insert and you won't be able to pull it out, okay? So you don't have to worry about ticks that you find on you that are questing or that you just caught right as they were inserting. But if you find a tick on you that is engorged and you're like, shit, I didn't know that was been there for like I don't know how long, then you start to wonder, okay, maybe I got a disease because maybe it was been there for a couple days. And the worst is if you don't even see it. Like the nymphs are so small, the larvae and the nymphs are so small that oftentimes you literally don't even know that they were on you. That's what happened to me with the Lyme disease. I didn't even know it was there until afterward. We already talked about this. Their saliva has all kinds of anti-immunity factors so that you don't notice them there, so that you can't feel it, so that they stick to you, all kinds of stuff like that. And there's actually a lot of people who study how the components of the saliva, the proteins in the saliva, might actually facilitate transmission of diseases. Okay, so sometimes what they're spitting in actually helps the disease become transmitted. And they take multiple blood meals to complete one life cycle. So 3 blood meals in the nymph, the larvae, nymph, and adult. Here's a female. Here's a male. These are, what tick is this? What tick do you think this is? Look at this. It's pretty orange. It's a deer tick. So if you look at, it's one of these. It's a deer tick. We already talked about this. I just say this again. The nymphs are so small that right now you think, oh, I could see that. But literally when you're like this far away from your hand, it looks like a speck of dust. Okay, so the way that they transmit diseases, I'm saying, is diverse, and it depends on the disease. Sometimes they'll spit it into you, so it has to get to the salivary glands. Sometimes they'll crap in you, so it's in their feces. Sometimes you'll smash it in. It's different depending on the different diseases. But in general, the longer they're on you, the more likely you are to transmit a disease. Okay, so that's the end of the tick section. Now we can kind of start talking about the Rickettsia stuff. Okay, so here's what you want to know about this family. Okay, so you want to know this Anaplasma Okay, this is a family of bacteria. Okay, and the characteristic of this family is they are all obligate intracellular bacteria. So we're going to talk a lot about this family because most of the diseases that ticks transmit that are bacteria, or not I don't know if I can say most, but many of them, all the ones that are famous are these except Lyme disease. And Borrelia is Lyme disease. So what does this mean? What's this mean? What? Can't live without host, but it's more than that. Like parasites can't live within hosts, but they're not obligate intracellular bacteria. So obligate means, you're saying obligate means that, yes. Obligate means they can't live without host. Intracellular means that they can only replicate inside another cell. So they're kind of like a virus. Can only replicate inside a virus, or inside a cell, host cell. So that would be a eukaryotic cell. So these are bacteria that live in your cells. So they're actually related to mitochondria. So if you look at where mitochondria came from, there's an ancient theory that if you look at the genes that mitochondria have, they're actually very closely conserved with genes of bacteria that are in Anaplasmataceae. So there's a thought, the theory is that the way that the first eukaryote got mitochondria is that it basically got infected with one of these obligate intracellular bacteria, and then they evolved a symbiotic relationship, and eventually it turned into a mitochondria. So that's actually a theory in biology. It's one of the leading theories of where mitochondria came from. So these are bacteria that literally live inside your cells. So that's different from something like E. coli that will replicate outside your cells. Okay, it's different. They can only replicate inside a host cell. Here's what the family looks like. So we've already talked about Wolbachia, okay? So we know what Wolbachia is. Wolbachia is one of these family members, but Wolbachia doesn't cause any disease. We care about Wolbachia for different reasons, because it manipulates the reproduction of arthropods. But these other things cause disease, okay? So anaplasma can cause human anaplasmosis, can cause bovine anaplasmosis. So you care about this if you work with cows. Ehrlichia can cause human ehrlichiosis. So these are all bacterial infections, okay? And it can also infect cows. Rickettsia is Rocky Mountain spotted fever, which we'll talk about a lot because it's one of the more famous bacterial diseases. And then there's one that we're going to talk about on a whole separate lecture, Orientia, which is scrub typhus. So these are all closely related bacteria, and one of the reasons they cause disease is because they have to replicate inside your human cells. So if they replicate in your human cells, that usually means that they end up killing your human cell, just like a virus. So that's why some of these cause disease. Okay, so history of Rocky Mountain spotted fever. The history of this is so interesting. I'm like compelled to erase this board. Hang on. So I can draw if I need to. Okay, so this is Montana. Missoula, so University of Montana is right in this town. And there's a river called the Bitterroot River. Okay? And this is, we first started learning about this disease in as soon as settlers started going into Montana. So late 1700s, early 1800s, okay? And before that, there were people living there. But the Native Americans here, it's not clear to me if they were immune to the disease because they had been here for so long or not. But before that, we started kind of hearing about this disease when settlers started coming into this area in Montana as we were expanding westward in the United States. And this is actually a figure from one of the first papers to study Rocky Mountain spotted fever, okay? And each of these dots is an incidence where they found a patient that had Rocky Mountain spotted fever, and this is where they lived, okay? So what do you notice about this figure? What?
It's along the river.
Close. It's all on the left side of the river. So it's all on the western side of the river, okay? So these epidemiologists, these very early people who are very early studying this disease, this is like late 1800s, early 1900s. They noticed this pattern that everybody who got the disease was always on the west side of the river, and you were safe if you were on the east side of the river. So land prices on the east side of the river were really high because everybody wanted to live here where you were not at risk of dying from this random disease. And on the other side, nobody wanted to live there because people were literally dying from this like random disease. So nobody wanted to buy property on the western side of the Bitterroot River because you're at risk of this disease. So this was a problem for when the state of Montana first formed, right? This is actually like a problem that the people would care about because land prospectors were like, well, I want to be able to sell this land for lots of money. But so we better figure out like what's going on so that we can stop it so that this land price will go up. Okay. So the government funded studies to research this problem on why on the west side of the river there was this disease. Okay. And the disease had very high mortality. It was 72% killing. So if you got it, you were more likely to die than to survive. And they called this Rocky Mountain spotted fever. So you'd get these rashes, and then you would mostly die. Let's see, anything else about this? So, let's talk about it. So, okay, so why do you think that it would only be on one side of the river?
It's where the forest is, or where the woods and stuff.
Well, ticks can't swim.
Like the woods are on this side of the west. That's what it looks like. I don't know.
No, it's not. It's not about the woods. It's that the ticks. So the ticks on this side don't swim across. Okay, and so I guess it's not that intuitive. So actually, what we learned much later is that ticks and this is still I guess quote unquote hypothesis for what explains this. But this is the best explanation. I guarantee you it's correct. So ticks, 8 legs, on the west side of the river are infected with a Rickettsia pathogen, which is Rickettsia which we now know causes Rocky Mountain spotted fever. And ticks on the east side are infected with a different Rickettsia, which is Rickettsia peacockii. And Rickettsia peacockii does not cause disease. Okay? And we've now sequenced the genomes of Rickettsia peacockii and Rickettsia, and we can compare them, and we know that this one has a lot of virulence genes that this one does not have. Okay? So we know why peacockii does not cause disease and why Rickettsia does cause disease. And if you survey ticks on the east side, you only find this one. And if you survey ticks on the west side, you only find this one. And you don't get much crossing of the river because ticks can't swim and the mice that they feed on don't swim. So there's literally this is like a case of like a geographical barrier. Like when you think about evolution or you think about like Darwinian speciation, the first thing that usually happens that's a requirement for speciation is I don't know what the term for it is, but it's like the geographic isolation. Yeah, there you go. So this is like a cool ecological case that's relevant to disease of where this actually happened. Like this river is a geographical barrier that the ticks don't cross. And on one side, these were ancestrally once the same organism, right? But then there was this barrier, and this one got some mutations in the genes that made it not cause disease in humans, and this one evolved some genes that did make it cause disease. And so this is literally the point where we had this geographic barrier that allowed speciation of the bacteria to speciate. So it's really interesting. And there's active research on, so if all the ticks on this side are 100% infected with peacockii, there's a theory that peacockii infection will exclude the infection of Rickettsia. So they're kind of like, even if you release some ticks in here, because all the ticks are infected with this, they would kind of like, they would like compete. They're competing for the same niche. So there's a kind of like an exclusion hypothesis. So that's the theory. That's the current theory of why in these old-time epidemiological maps you only found cases of this disease on the west side of the river. Still got 5 minutes. This is what the land looks like. So you can see, it's beautiful there. So you can see why like the landowners were like, we got to get this figured out because people should be buying this for like high prices. Okay, so we'll talk about this guy. So all of Rickettsia, the genus of bacteria, is named after this guy, Howard Taylor Ricketts. Okay, so he's a famous hero who was one of the early people to study Rocky Mountain spotted fever. Okay, so in the early 1900s, there was considerable debate about the cause of Rocky Mountain spotted fever. People didn't know if it was associated with ticks. People didn't know that it was associated with a bacteria. That was all yet to be discovered. So there was this group of people, Wilson and Chowning. Okay, they were a group of researchers, I think they're from Mayo, who went out there and they did some studies, some field studies, and they came up with a hypothesis that the ticks probably transmitted some kind of protozoan. Okay, so this is right after Patrick Manson had discovered filarial worms, can spend a portion of their life cycle in mosquitoes. So again, I was saying that, and when I originally gave that lecture, I was saying there's kind of like a big bang of like explosion of thought, where then people started thinking, well, what about this disease in my location? Maybe that's spread by arthropod. Okay, so then these are the first people to kind of take that thinking and go to Montana and start testing the ticks or the arthropods in the area that were sucking blood. And so they came up with the first hypothesis, but they were wrong that they thought it was a protozoan. So they thought it would be something like malaria, whereas in fact, it was something completely different, a bacteria that only replicates inside a cell. So that means also, if you're looking for bacteria that only replicates in the cell, it's going to make it harder to find than necessarily like a protozoan that's just bigger and sitting in the blood. Okay, so this was in the glory days of when you just go out and you find some insects and you smash them up and you literally just put a chemical on them, which is gimsistain. Gimsistain stains bacterial outer membranes, and then you just look for stuff. You look for abnormalities. So this is like all they had to do to get a genus of famous bacteria named after them. It was a little more complicated than that, but it was a lot easier to find new stuff back then. So this guy, Ricketts, so this is after Wilson and Chowning. It wasn't yet resolved. They thought there was the ticks maybe in the process. This guy, Ricketts, decided to go to Montana, and he actually spent his own money to do it, okay? So this is a guy who was so interested in science, he was literally like, I'm going to go spend my own money, and I'm going to study this, which is fascinating. And the energy is cool. So he's going to do an independent study on Rocky Mountain spotted fever. So he discovered quite a bit. Ricketts was the first person to identify an animal that continually passage the disease. So if you ever want to study a disease, you have to have a way to grow it, right? And one of the problems with the obligate intracellulars is that at this point, after like Pasteur and Cox, everybody started just taking solutions and trying to grow bacteria in like broths, okay? So the obligate intracellulars, because they replicate in a host, will never grow in a broth, okay? They will not grow. So one of the early ways that was defined as a way that you could characterize a new microbe is you had to be able to demonstrate that you could grow it. Nobody could grow these because they only replicate in host cells. Okay, so he was the first person to figure out how you could actually grow an obligate intracellular, and all he would do is he had guinea pigs, and he would inject them, and then they would get sick, and then he would take some of their blood and inject it into a new guinea pig, and he would just passage the disease in guinea pigs. So he figured out a system through which he could study Rocky Mountain spotted fever by infecting and passing it in guinea pigs. Where's time? Okay. We'll stop here and we'll continue with Ricketts the next lecture.